Good afternoon, everyone. I am Jim Sluka. I'm a senior scientist in the Biocomplexity Institute. I've been working with Professor Glazier for about 15 years now. I'm a computational chemist and a computational biologist and kind of a hardcore CompuCell end user, much less involved in the developmental side. We're going to talk about a whole bunch of things here. This slide is a little out of date. We talk a little bit about best practices for parameterization, initial cell layouts, how to set up your initial configuration, and then hopefully a little bit about parameter scans. There I am on the list of all the people. We're going to start off with some hints and tricks. We're going to go through this kind of quickly. I can share the links to the slide deck and my demos. Put that into chat. There's two links there. The first one's to the slide deck, and the second one is to a zip file that has a bunch of zip files in it of CompuCell projects. Back over here. So hints and tricks. We do this kind of quickly, just some random bits and pieces that might help you be a little more successful in your modeling. The first one is when you define the number of steps, back up, add one to it. Always do one more step than you think. This is useful because you frequently do things every 10 Monte Carlo steps, or you're saving screenshots every 100 Monte Carlo steps. You want to make sure that when your simulation shuts down, that last set of things that you wanted done gets done, so that you don't lose your last screenshot or your last analysis. So just add one to all of your target number of Monte Carlo steps. The other thing is you should pretty much always change in the pot section, change the neighbor order to 3. That'll reduce pinning and your cells kind of always looking very square and blocky. In your XML, you can tell CompuCell to use more than one processor. You can use as many processors. You can ask for as many processors as you want. If the number is too big, CompuCell ignores it. We'll use whatever is available. But if you're going to use more than one processor, you really should check the timing report and make sure it makes a difference. Often, one or two processors is all you can use. You can use more processors. It'll use more CPU time, but it won't run any faster. So you can check your performance report that you get at the end of a CompuCell run. Currently, there's a bunch of debug output after this report in your command window. So look in the command window and scroll up and find this report and see if changing the number of processors actually changes the total runtime significantly. The next thing to think about when you're thinking about getting it to run fast is in this report, it tells you how much time was spent in every steppable in your code. In this particular code, 18% of the CPU is spent in a steppable. You typically don't want your steppables using very much time. 5%, 10% is okay. But in some cases, if your steppable really is doing more than it needs to, and this number starts to be 50% or more, you probably want to explore your steppable a little bit, see if you can get the steppable to run a bit faster. Usually this is caused by doing something every MCS. Some steppable calculates surface area every Monte Carlo steps. You rarely actually need to calculate stuff that fast, that often. Same thing with growth. You rarely need to increase the cell's target volume every Monte Carlo step. You might as well do it every 10 Monte Carlo steps and just give it a bigger growth step when you do that. So look at your steppables, try to keep this number reasonably low so that you're not spending all of your time in steppables. So there are a number of ways to change how often a steppable runs. You've probably seen some of this in the project Pi file where you register your steppables. There's a frequency parameter. In the class definition itself, inside your steppables file, there's a frequency parameter. I think most people tend to use something where they just actually put it directly into their code. So here's a step inside of a steppable, and it's going to do some code every 10 MCS. And something else is going to do every 100 MCS. and something that's going to do only between 1 and after Steppable 0, excuse me, after Monte Carlo Step 0. It's going to do it every 10 step. There might be something in the first Monte Carlo step that's not defined yet. This seems to be the more common way of controlling how often you call a steppable. The next thing is decide on some basic units for your model. It doesn't matter if you change it later, that's perfectly fine. But at the very beginning of your modeling effort, you should decide how big a voxel is. A voxel is 1 micron, that's fine, great starting point. Decide how big you think a Monte Carlo step is supposed to, how much time, real time, simulated time, the Monte Carlo step is supposed to represent. 1 minute per Monte Carlo step is a reasonable starting point. In some cases, the Monte Carlo step might be a day, it might be a second, but in any case, you should make a decision early on what you think that these two fundamental units are. You can always change that later. You can always change how you interpret it later, but write down somewhere what you think it should be. One way to do that is at the top of your steppable file. So here's the first line of a typical steppable. Just go ahead and put in a multi-line comment and define your basic units. 1 voxel is 1 pixel, is 1 micron. 1 MCS is 1 minute. This has no programmatic use, but it does mean that you've committed to some understanding of how big these basic units are. That was the same thing. So next thing, trying to keep things organized and tracked. Every time you run a CompuCell simulation, you get an output folder. Every time you tweak your code, things have changed a little bit. So what are some good ways of kind of organizing your workflow and keeping track of what you tried and what you haven't tried yet? Programmers like to use GitHub or SourceForge and do official versioning of their code. If you're a programmer, you're probably already very familiar with SourceForge and GitHub and similar tools. It's very robust versioning. There's always an old copy of previous versions of everything. It just has excellent ability to share with others. Other people can download your GitHub, can clone your GitHub, can modify your GitHub. Disadvantages of it, non-programmers usually have not used these tools, and the fact is they're not easy to use. It requires a significant amount of learning to properly use a GitHub repository, for example. In addition, it requires you have internet access, which isn't usually a big deal, but it is something to consider. So that's one approach to GitHub. The second approach is to just version into a Google Drive or Dropbox or some other repository where you can put a whole bunch of stuff. So it's easy to do. It keeps a reasonable set of backup versions. Google Drive, for example, will keep back previous versions of your projects. It's reasonably easy to share with others. It has some disadvantages. It can be tricky to use on your local computer. There's sometimes some confusion about, can you open a Google Drive from Python? It can get a little tricky. Again, it requires internet access, and cost may be an issue. CompuCell simulation files are pretty small, but simulation outputs can be quite big, especially if you're saving a lot of screenshots. So it's not unusual to get into the hundreds of megabytes of data from a simulation. And now you got problems with how much quota do you have on Google Drive or Dropbox, or do you have to, pay for more storage space? Non-programmers tend to not keep old versions of code. So that's an easy thing to do. Your system may have automatically generated backups, but it's probably not a good idea to count on automatically generated backups to keep old versions of things. So there's a little protection from file loss, little ability to trace the kind of the evolution of your code. It's easy to develop broken code that you can't revert back to the last version that was working. And it's not easy to share if it's kept locally. A reasonable, low-cost, simple thing to do is just simply keep zip files of your project periodically. So you can go to the top level folder, right-click on it, create a zip, and then name that zip with a timestamp. 2025-08-06 is today. And just keep those zip files around. If you're doing just the code, those are very small. You can also do that, the zip files of your project outputs, which will tend to be a lot bigger. But that's a fairly easy way to keep, here's my version of such and such date. Here's my version from the next day. Here's my version from the day after that. The big advantage is you actually have to remember to do it. If you don't do it, you don't have them. So as I said, an easy way to do it is, this is the CC3D workspace. So I have some output folder that was created when I did a simulation run. Inside that folder are all my simulation files. It also has all my screenshots if I was saving screenshots. So you can just right click on this folder and make it a zip and put that zip someplace handy. Always a good idea, at least once a day. Make a zip for that day and save it somewhere. The next thing to think about is, we already talked about, somewhere in your code, define how big a pixel is, define how big one Monte Carlo step is. You really should be keeping a Word document or some other editable text document that'll handle figures, that keeps a description of the basic biology you're trying to model. It should have a short description of the domain. It should have a list of the biological entities you've included in it, what processes are there. Often, a simple diagram is the most useful thing you will have. This is a diagram that a collaborator at NeighborOrder sent me 2 years ago about modeling follicle implantation in the uterus. And this was the beginning of my model. And this figure is always part of my documentation for the development of the model. This is where we started. It lists the cell types and kind of the overall flow of the process. So as I say, keep it as an evolving Word document. It could be kept as a plain text file inside your code, which is also not a bad idea. And we'll talk about that a little bit more later, I think. But keep that file somewhere. If you put it in your project folder, it will, so in my, wherever your working folder is, if I put a Word document in that project folder, that Word document will be copied to the workspace every time you do a run. So if that document's in the right place, it'll version and go into your output files. Okay, keeping track of parameters, notes, etc. Within a computational project, parameters can really get you confused. Often you have biological parameters like cell volumes or diffusion coefficients that are not anywhere in your code actually defined. It's some random variable name that's not labeled and it's not specified. Often these biological parameters are scattered about in your code. Your diffusion module has a diffusion coefficient in it. You have to go hunt that down if you want to document what the diffusion coefficient was, or if you want to try to change that thing. So models should try to track and organize their parameters and clearly label parameters that are biological in sense. Oops, can't click on the screen. Versus parameters that are purely modeling parameters. So a biological parameter would be a cell volume. A modeling parameter would be that my cells are 10 pixels wide. And those two things are related to each other, but you should document them separately. So again, one way to do that, at the top of your Python Steppable files, you can just put in and make these definitions as code. So here I have an example of them. I'm going to explicitly say a voxel is 1 micron in width. Now, when I do things like scale the model, I can use this voxel to micron conversion factor to define how wide the model is. If you define variables outside of any class in a Python file, these are global variables. Any Steppable can reference these variables. You can also use CompuCell's global dictionary. One advantage of doing it this way is individual modules can't change these values unless you use the global keyword. So these things are basically frozen. So a better way to do parameters, though, is to separate, instead of putting them in your code somewhere, bunch of comments, some values defined. a better way is just to go ahead and break the parameters out into a separate Python file. And you can include that separate Python file in your CC3D projects. So in that parameter file, you can make your basic definitions like one pixel is 1 micron. You can include units, at least as comments, and you can include references for where you actually got the parameter. Put a citation in as a comment. And you can include ontological links as some examples here. A lung epithelial cell has this particular OBO library ID. So to do that, it's fairly straightforward. In Twedit, you can right-click over here in your file list and tell it to add a resource. That'll open a dialog that asks for a file name. You can point it to a local file. A file type, in this case, we're going to use it's a Python file. It should live in the simulation directory for the project and the project name. These two should automatically be filled out correctly. You hit finish, and now you have in your file list, you have a new file called parameters.py. Of course, you could have named it anything you wanted. And in your file tabs, you've got a new parameters.py file. So in that file, you can do things like, so I've got a comment and a date that was created. I've got the model, some things defined in biological units. I want my model to be 10 cells wide. I want an individual cell to be 10, and I've got units. So width is in cell widths, and this number is in microns, and a little comment. I got my pixels per micron conversion factor. And then I can do math and various other things with those values within this Python file. So here's that same file again, zoomed up a little bit. And now I can do things like my model width in pixels is the model width in number of cells times the cells width in microns times the pixels per micron. And I can look back up through my little table here and find out what my units are and things like that. And I have all of my stuff, all of my parameters in one place, and I have the capability of calculating parameters based on other parameters. And I think we'll try to do a demo later on showing how this would be used. In this case, the goal was to be able to change one parameter and change the width of the model in pixels without changing the width of the model in the number of cells that are actually in the model. So I can have little cells and have the model 10 pixels across, or 10 cells across, or have big cells, 100 by 100 pixel cells, and still have the model be 10 cells across. So this is a good way to keep track of all your stuff. When you're doing that, how do you actually use it? Well, now you can go into your parameters.py file, into your Steppable file. So here's the top of the Steppable files, and I can import my parameters file. So by default, it will take this name and look for other names in the project and find this parameters.py file. Notice there's no extension on, it's not parameters.py, it's just parameters. And I give it a name, a local name for the domain, for the variable namespace. And now I can reference things in my parameters file just as p dot and then whatever the name is in the parameter file. So here, excuse me, here I show setting a cell target volume based on some parameter that's in the parameters file. As I said, so this is just an example of in a Python-only simulation, I can actually use something in the parameters file to define the width of the model in pixels. So this value is calculated in the parameters file. This only works if you do a Python only. You can't use this trick to change things in the XML, at least not easily. There are some ways of doing it, but it gets a little trickier. But if you're doing these global parameters and you're trying to keep your parameters well organized, it's a good idea to do your simulation as all Python instead of a mix of XML and Python. So as I said, this is an approach of defining your model in the number of cells across the window, and then the cell width in microns, and then conversion of microns to pixels. So here I'm calculating the total width of my CompuCell model in pixels as being a function of my earlier values. That's what I just said. So calculating something based on my basic parameters. And again, here this is the Python version of the XML, so I could replace this hard-coded model definition with a variable name. If you want to do a Python-only simulation in Twedit, that's one of the options very early on when you are defining a new model, tell it Python-only. In the zip file, I put a link into, if you open up that zip file, it's got a bunch of zips inside of it, and this might be the wrong module number, but there's an example of parameters if you'd like to open up that project. So as I say, if you click on the Python only, your XML file instead gets created as a Python file, which means you can import your parameters file, which means you can do math as you're assigning what normally would be done in the XML. So I'm looking for a demo CC3D parameters example. This one. I'm going to unzip it to my desktop. Actually, I'm going to do it manually because it's a little bit quicker. Put it on the desktop. Start to quit it. Take a moment for Twedit to think about it. Might as well make this bigger. So as I said, so this is a Python only simulation, so there's no XML file. And this is the Python equivalent of the XML. You see we have import the parameters as P, and over here is my parameters file. Calculating the model width in pixels, the number of cells across the model, and it's also rescaling, attempting to rescale the adhesion values. This is a cell sorting model. Import P, and here is assigning my model based on the thing calculated, the dimensions in pixels based on calculated in the parameters file. And here I am assigning my adhesion values based on values that are in the parameter file. I can actually run this, and it should run. Haven't tried it recently.
We do have a good question in the chat. Is there an advantage to using XML over Python only?
The advantage of XML, first of all, that's what CompuCell was originally written in, and it's possible to write a simulation using just XML and no Python at all. I think in practice, you almost always have XML plus Python. So I am leaning towards might as well do everything in Python because it's a lot easier to change things in Python than it is to change things in XML, particularly programmatically. So it's really tricky to change the model definition in the XML programmatically. It's much easier if all that information is already in Python. So this simulation is running, and it's plotting the heterocontact area between the condensing cells, which are the blue ones, and the non-condensing cells. This particular simulation, though, you have to go in the back, look at our parameters, what our J values were. It is slowly sorting. You can speed it up. You can do limited steering without actually defining steering. I can go here to pots and find the pot's temperature, make it really hot. It's still going to take a while. Cell sorting can take quite a while. Let's cool it back down to where it was. Okay, let's see, where's my PowerPoint? So this is a simulation running. If you let it go long enough, eventually it will sort in this rough metric of the contact between various cell types will drop down, indicating that the cell sort was successful. I probably should stop that. It's going to keep chewing along. I'm going to skip over this. This is just how you scale adhesions and lambda volume, lambda surface as you change the model dimensions. This is an example of changing just this one parameter, pixels per micron in the simulation. Pixels per micron is 0.2. The simulation is 40 by 40 pixels. Change it to 0.5. Now it's 100 by 100 pixels. Change it to 1, and now it's 200 by 200 pixels. So this model is much coarser, runs much faster. It exercises all of your code, but it might not actually be giving you a result that you want to use. It just is an easy way of testing. Does everything work without having to wait a very long time for simulation to run? So that's one of the advantages of using a parameters file. It lets you rescale models, it lets you collect up your biological parameters, your CompuCell parameters, define units, make notes, make explanatory and to-do notes and things like that. Some caveats on rescaling model is some things are kind of tricky to do. One use of rescaling a model is to establish that your model's behavior is not dependent on how big you say a pixel is. If my cell is supposed to be 10 by 10 microns across and I model it as a 3 by 3 pixel cell, do I get a different result if I change that to 10 by 10 pixels per cell? The fact is it will, up to a point. If your cells are too small, they behave differently than if too small in terms of number of pixels. They behave differently than they will if the cell is bigger in terms of number of pixels. But by studying how the model behavior changes as you change the size of a pixel, you can usually establish that at a certain point that there is no difference between, for example, 20 pixels wide and 25 pixels wide, except for the 25 pixel wide takes 20% longer to run, but you get the same results regardless at some level of resolution. So best practice logging the parameters. If you're going via the parameter file, every time you do a run, that parameters.p file will be in your simulation, will be copied to the output simulation directory. So if you ever go back and pull up an old simulation result, your parameter file is in that folder with the simulation, so you know what parameters were used to actually run the model. There's a lot more complicated ways of doing this. In this example, what I'm doing is I'm using a dictionary so that I can have multiple sets of parameters in my parameter file. So I create a parameter set, and I have this dictionary of its characteristics. So it's pixels per micron. Its value is 1. Its unit is pixels per micron. Its reference is not applicable. I don't have a reference for it. I just make this dictionary. Then you can use the typical Python stuff for making nice, pretty outputs of a dictionary. If I increase the number of layers in this dictionary, which I don't think I show, if I increase the layers of this dictionary, I can have multiple parameter sets and have them all in the parameter file. So this is an example of experiment one. If experiment one, here are all my parameters I want to use for experiment one. If I set my experiment variable to 2, these are the parameters I want to use for experiment 2. So in one project, I can have the variations on parameters that I've been looking at. Oops, I can't use the scroll wheel. So this is a way of having one simulation that has multiple parameter sets where the parameters are well defined, and you can change one variable in the model and have it pick a particular set of parameters to run. So some conclusions, this first part of the talk. So best practice, establishing a consistent way for tracking your code. At least copy your project directory, zip it, put a date stamp on it, and keep it somewhere. The absolute minimum. If you're up to it, go ahead and go to something more complex like Google Drive or GitHub or something like that. But whatever you do, do something. Don't let your model just slowly wander about and lose track of what you've tried and how the model has evolved. Make sure you're making at least some minimal effort at tracking the parameters in your model. Have a clear definition of how big a pixel is and how big one Monte Carlo step is. If you're pulling information out of the literature, some paper says that a hepatocyte is 20 microns across, put that into your your tracking process, either in your Word document where you keep track stuff, or better yet, in your parameters file, say that this value came from this paper. It'll be much easier down the road, one, to present the model, and two, to try to write up a manuscript describing the model. Let's see, there's some comments. My comment screen is way over here. Yeah, Michael made a comment. In terms of periodic boundary conditions, so speaking of diffusion coefficients, how does CC3D keep track of properties when using periodic boundary conditions? The properties don't change with periodic boundaries. The diffusion coefficient is independent of the behavior at the wall of the model, whether it bounces off the wall or wraps around as it does in periodic boundaries. So Michael made some answers. As I think I addressed, is there an advantage using XML over Python only? Hayden responded that XML is a little more readable than is Python code, which is true, but XML is really not all that easy to read either. And it's really not all that easy to edit. It's really easy to mess up XML code, not close a class or something like that. Okay, so that's best practices, kind of a start. The more you do the best practices at the beginning of your project, the better things will be when you're 2 weeks into it. So you've got the CompuCell simulation that shows that has parameters file and some definition of units and things like that. Feel free to play around with it. You can change that one parameter that controls the conversion between microns and pixels, and make little cells, make big cells, and kind of explore that code. Now we're going to talk a little bit about initial cell layouts. So how do you get to initial configurations? Throughout the course, I'm sure you've seen some of the tools. There's in Twedit, when you create a new project, you can use the Blob Initializer, and it's one of the things that you select when you tell Twedit what you want to include in your simulation. Same place where you tell it you want to include the volume constraint, things like that. There's a blob initializer, uniform initializer, tube initializer, polygon initializer, and I'm going to show some demos of all of them. Surprising, here's links to the documents online. So here's a quick example of kind of using them all. Actually, I'll take that back. This is just using uniform initializer and blob initializer together. There's a demo, slab blob demo in that zip file. Here we use this little piece of uniform initializer. It's labeled A, created this box down here. It's using multiple cell types, and the proportion of the cell types is given by this list of cells. So I've got a whole bunch of A cells and much fewer B cells. And that proportion will be mimicked. I have a gap defined so the cells don't actually touch each other. There's the cells are 5 pixels wide, and the whole thing is a box with these dimensions. So that this A code here creates this code down here. B creates another one. In this case, it's only C cell type. And this happens after this one. So these new B cells overwrite the existing A cells. And then we make a region C, which started off as a green solid section. But then in another command, we make a smaller box of medium, which basically erases all the cells inside the box. So now we have an empty box of cells. Uniform initializer makes rectangles. Blob initializer makes circles. So you do the same kinds of things. E is this big region. It's got gaps defined. So there's gaps between the cells, and it's an equal mix of A cells and B cells. The region F is this central region. And again, this one's a single cell type, but it was called after the first call. So these red cells overwrite the existing set of cells. And then G kind of does the same thing, but offsets a little bit. And these blue cells overwrite some of the red cells and some of these original cells. So you can often pick up and make fairly complicated patterns by using a combination of uniform initializer with multiple regions and blob initializer again with multiple regions. So as I said, later regions overwrite earlier regions. So that's one approach. If you can do it as a circular region, a rectangular region, you can fill an area with one cell type and then overwrite it with another area of a different cell type. And doing those, mixing and matching these, you can come up with some pretty complicated patterns. But there are even more useful and more complicated ones you might want to use. Polygon Initializer, there's some demos in the zip file, lets you define the points in a region and then fill in that region with cells. So here we're defining, we start from X10, Y90, which is here, and we go to 1010, which is this edge, and then just work your way through all the points. And then ultimately say, fill that polygonal region with cells of a cell type. In this particular case, there's no gap, so the cells all touch each other. And this works in 3D too. It's done a little, the z-axis is done a little bit differently. It's done as an extrude, so you define a shape in 2D and then extrude it into 3D. Tube initializer, again, it's similar. It lets you generate tubes of cells. Again, if you click on this when you're building your, when you're choosing your plugins in your model, if you click on Tube Initializer, you'll get some boilerplate code in your XML, and you can modify it as needed. In this particular case, I defined this green tube first, and then fill it with this red tube. And the red tube is only one cell wide, and the outer green tube is multiple cell wide. So you get a tube of cells, a tube filled with a single cell type. Another way of doing it is you can, in a simulation, you can save PIF files. A PIF file is program information file. It was kind of an arbitrary name, but it's an exhaustive list of every pixel in the simulation and what cell that pixel belongs to, what fields are on that cell, if it's a compartmental cell, what compartmental cell that belongs to. So in your Steppable, you can load a PIF file and start off with a particular configuration, which could be quite complicated. Within a CompuCell simulation, within Player, you can save a PIF file. So if you've got some simulation that you need to run for 1,000 steps to kind of let it equilibrate and settle down, and you don't want to have to keep rerunning the first 1,000 steps, you can run it to 1,000 steps, save the PIF file, and then go back into your XML and tell it from now on, just load this initial PIF file. In addition, you can generate a PIF file by hand. The format is fairly straightforward. It's just a series of lines. The first number in the line is the cell number, and then there's a cell type name, and then there's X minimum, X maximum coordinate, Y minimum, Y max, Z min, Z max. You can do these each line for each pixel, or if you have rectangular regions, an individual line can define a rectangular region. So you can write a Python script outside of CompuCell that would generate a PIF file if you have something very complicated that you would like to generate. You might generate it from an image, from a cartoon, from a histology slide, from some other source. But you can generate a PIF that you could then read into CompuCell as your initial configuration. On top of all that, so those are kind of the automatic ways of doing it. You can manually create cells. If you can write mathematical code, mathematical equations and Python code to generate a region of space, you can make those regions into cells in CompuCell. In your Python, there is CC3D Python, cell manipulation. You can create a cell and create a cell detailed. I'll skip the simple one-liner one and just go, this is the create cell detailed. It'll paste in some boilerplate code that basically says define the cell type. So type name here will be replaced with one of your cell types. Give it an X and Y bottom left corner and then a size. This is just an example. And then you say, you assign your new cell to this region in the cell field. This region can be a region. So in this case, X starts at X, which is the X position, and its width is X plus the size minus 1. It could also be single pixels. So it could just be X, Y, Z, and you're assigning that single pixel to this cell. At any time, you can modify the cell field and change a pixel to some other cell. So I've got some cell 2 at someplace else in the simulation, is some other region of space in the pixel field. Its cell ID is this, and I can force that cell ID into the cell field. So I can add any pixel I want to another cell. The tricky one is you can make a pixel into medium, and the tricky part is that there's no cell type medium. So you've assigned the cell field to the Python quantity called none. So you can create your cells, you know the shape, you can create a big cell and then go in and delete pixels in the cell basically by assigning individual pixels to none. If you're creating cells, remember, if you define your cells in the XML or in the Python equivalent of the XML and you say cell type A has a target volume of 10, every time you make a cell type A, it'll get assigned a target volume of 10. If you define all of your target volumes in the Python and you use kind of the simple, you define the plugin in the XML, but you don't actually assign anything to the target volumes, then when you create a new cell, you have to remember that you have to set the target volume for every cell target volume, target surface, lambda volume, lambda surface, whatever plugins you're using. but you need to assign all those things to the cell you just created. And an easier way to do this would have been to do. I created the cell of the right size, so my target volume is whatever the volume of that cell was I just created. So there's some other, here's an example of some Python code. What it does, it makes this ring of cells, and there's 4 cell types. They're all the same size, but they're generated in this circle. This is in the demos that's in the folder. So basically, like I say, if you can write mathematics and code that can generate pixels, you can assign your cells using that list of pixels. I just sweep around an angle which goes around the circle. At every point in the circle, I make a square box. I check every pixel in the square box to see if it's within a particular radius of the center of the box, which is what clips it to be a circle. And then I assign all those pixels to a new cell. One thing you have to be worried about is if you're mathematically generating your pixel coordinates, CompuCell will not check that your pixel coordinates are valid. So you can, your math might say pixel -5. Well, there are no pixels at x -5. There's no negative numbers in the pixel coordinates. Or you might generate a pixel that's bigger than the dimension of the model. CompuCell will not check that. So you have to make sure that when you're creating your pixel lists, that they're actually within the model. You can do that with, this is the global dimension of the model on the x-axis and the global dimension of the model on the y-axis. And these values are available in the Twedit pull down where you get model parameters. So if you forget what this variable is called, you can get it from the pull down. If you really want to go whole hog, you can generate your cell layouts from an image. There's a demo in the zip. For example, the demo takes this little section of a H&E stained histology slide and imports it into CompuCell. CompuCell, we don't have control of the color bars, so I can't redraw the image the way it was originally because I can't assign individual colors to a pixel. But this is various channels that are created, or various representations created from the various color channels in the original image. I could then write code that would take this image and segment it. Should be tricky, but it could be done. And that could be your starting point for some simulation. So there's a demo for that. There's also a demo from the front page of CompuCell for how to import an image and use that as a starting point for a initial configuration. I'm not going to have time, so I'm not going to go through this in detail, but there are slides here about how you actually go about it. It has to pretty much has to be a bitmap image and not a vector graphic. Unfortunately, this particular library that I use doesn't work with GIFs. So the usual caveats is you got to find a Python library that'll handle the file format. But you can, in CompuCell, you can load any Python library you want. So you can import the PIL image library or the CV2 library, which is a more modern image processing package for Python. So you can import any packages into the system you want, or into a CompuCell simulation that you want. So just tons of detail about how they actually take the image apart and put it into pixels in your field. So we went through really quick initial layouts. So using the various built-in initializers, Blob, Uniform, Tube, and Polygon. The idea that in general, you can stack them on top of each other. And whatever the last assignment for a pixel is what will end up as your starting configuration. And using a combination of these, you can usually get pretty complicated layouts. You can use PIF Initializer either with a PIF that you created externally CompuCell, or by saving a PIF during a simulation, kind of a setup equilibration simulation, and then starting all your subsequent simulations, starting from that particular initial configuration. Talked about manually making cells in Python, and also how to import an image and how that might be the beginning of creating a cell layout based on an image. Hayden, I don't know if we have time, and if people want to try to use all the various tools to create a layered model, or if we should go on and spend more time trying to do the parameter scan as a demo.
What do you think, James?
Well, they're both worth doing. I think, but how much
We can,
time?
yeah, it's. I got about 35 minutes left. I think we can do this pretty quick if people want to follow me along.
I think so. I think
Yeah, I
it's
think
worth
that'll be
doing.
good.
We can at least get, kind of the workflow going, and you might not get the details correct because sometimes it takes some fiddling. So let's try that. So let me get my Twitty going.
I do have one question in the chat. Is it possible to import a black and white file, like a TIFF file, to directly initialize a particular cell type?
Yes, you could. And you can also, there's a demo, and it's kind of painful because it's kind of old, but let me go to the front page of CompuCell. If you come over here, there are visual examples, which are just meant to be, I can explain what it's doing by showing you a picture. So here are some of them: track a cell location, etc. But here is an example of this. Was drawn in PowerPoint. And the idea is that ultimately the blue regions will be inside a blood vessel. The green region are cells. The little red things create blood cells. And as they get created, it just pushes blood through this pattern. So I created this in PowerPoint and then imported it into CompuCell. The nice thing about creating it in PowerPoint is you know exactly what every pixel color is. There is only one color of green. There aren't 14 shades of green in this file. So when CompuCell looks at a pixel and it sees this particular green, it knows it's supposed to be a particular cell type. And it knows that, well, I told the code that black means that's separation between cells. So every time you go across the border, you get a new cell. So you could use a black and white image. You could use a color image. If you create it yourself and you have perfect colors, say there's only 4 colors in the image or 5 colors instead of being 16 million possible colors. It's reasonably straightforward to read that into your Python, convert it into a cell field, convert it into cells, and use that as a starting configuration. Okay, so we're in Twedit. What we're going to try to do is this: create these cells, parenchyma, matrix, basement membrane, and epithelial as a stack. Okay, so we'll have parenchyma and matrix on the bottom as a bigger region, and then a layer of basement membrane, and then a layer of epithelial, and then a thick layer of medium on top. So we're going to do a new project. I'm going to put it someplace more useful. Close enough. I'd actually create the folder first. Let's see. Let's do, since we're using the initializers, we'll do Python plus XML. 256 by 256, that's fine. We are going to use, we want demos of both. Unfortunately, the initializer only lets you use one. Actually, rectangular is going to work. We don't want blob. We want sheets of things, which are by definition a rectangular and not circular or spherical. None of these matters, but it's always just a good idea to make the pixel copy range bigger. We need some cell types. I think we had just someplace else where I can see it. You won't be able to see it. We had parenchymal. We had matrix. We had basement. And we had epithelial. We don't need medium because that's already defined. Now we have our 5 cell types. We don't have any diffusing fields. We're not actually going to do anything with this model, so I'm not going to load a whole bunch of stuff. So here's our Blob Initializer. So maybe I can do this so we can see. So we want a region of parenchyma and matrix as a thick bottom layer. So our model is 255 pixels, 256 pixels wide. So X is going to go all the way across the model. Oh, that's not right. This one should be 255. This starts at the bottom left corner. Model, so let's make it 50 high. No gaps. Say there are 5 widths. The helper put in all our cell types. This is supposed to be the parenchyma and matrix only. And let's say that there's a lot more parenchyma than there is matrix. That's our first layer. And then for our second layer, we're supposed to do a basement layer, basement membrane. I'm going to come up here and steal the variable, the cell type name. So this is only going to be these guys. It has to sit on top of this previous box, but it will still go the full width of the model. So X from 0 to X255, but it sits on top. So we'll make this 50, and we're going to make this thin. So it's only going to go to 52, and I can never remember if I need to have 1 extra or not. And we'll say it's only 2 thick. Okay, so that's our layer of basement on top. We want a layer of epithelial on top of that. It'll look very similar. Steal my epithelial name. Still goes from X0 to X255. It has to sit on top of this previous layer, so we'll make this 54. And we'll make these cells bigger. We'll make this 60. It should be all the changes we need. What it does. So it looks pretty good. Here down here, if we look at our cell type name, so green and blue are the parenchymal and matrix. We have a lot more parenchyma than we have matrix. The distribution is random. We have a thin layer, a basement layer, thin basement layer, which looks like it's 2 by 2, which is not quite what we wanted. Then we've got the epithelial layer, which came out as 2 layers. So my boundaries are not quite right here. Let's see, this is the parenchyma underneath. This is only, so actually it's 52. There we go. There we go. 1 layer. Got this blank layer because I didn't move down. When I made the red layer thinner, I didn't bring down the yellow layer on top of it. So this one ends at 52. Actually, this one needs to start at 52, and it's supposed to be 5, so it's 57. There we go. So a big blob of parenchyma, a thin epithelial layer, and then some epithelial layer cells on top of that. And then we didn't define anything, everything kind of default parameters. So this model is probably going to fall apart. It's assumed some target volume. So that's one thing I didn't take into account. So for instance, my epithelials, I've got to be, I want to be 5 by 5. But up here, epithelials has a target volume of 50. That should be 25. And our basement with 2 by 2 or 4. How big did we make these? We made these parenchyma and matrix 5 by 5, so that's also 25. Now at least the assigned values for the target volumes roughly match the values that the model starts with. My basement membrane disappeared. Anyone want to venture a guess about why my basement membrane disappeared? It probably disappeared because the adhesion values are relatively large. Where's my adhesions? They're all 10s. My lambda volume for these little cells is relatively small. And it's just randomly, the random walk is just deleting the cell, and adhesions are swamping it out. So I can make sure these cells persist, making their lambda volume much bigger. At 20, we'd probably almost freeze it in place, though they probably will still wiggle a little bit. Let's try it with a much bigger lambda volume. Nope, still not enough. They're persisting. Actually, they're persisting. They are getting squished a bit. Now what's happening is all of our adhesion energies are the same. So we haven't told the model that the basement membrane should stay between the parenchyma and matrix and the epithelial cells. So the cells are just wandering about since we have no regulation on where they should be or where they prefer to be. We can freeze the basement membrane. This will probably freeze, give a really big lambda volume so that there's a huge penalty if you do anything to the pixels of a basement membrane. So now they've stopped moving. So the more fundamental problem here is that we need to assign our J value so that we maintain the parenchyma and basement membrane epithelial layer. Okay, so that's showing this. We're just using the uniform init, multiple regions. You can generate reasonably complicated patterns. If you start mixing in the Blob Initializer and some of the other initializers, you can usually get a starting configuration that you like. If we were doing this as a circular field with the blue and green in the middle and then a layer of red and then a layer of yellow, we could use the Blob Initializer and start off by making a really big region of the blue and green, and then a smaller region of red inside that, and then a smaller region of, actually I did it backwards, a big round region of yellow, and then fill in with a smaller round region of red, and then fill inside that with a round region of blue and green. We start off yellow, replace it with reds inside, then replace it with blue and green inside of that, and get the nested pattern in a circular layout instead of this horizontal layout. Okay, how long did that take? 10 minutes, that's not too bad. This slide deck, if you download the slide deck, has a lot of other things in it. So there's some slides talking about how you write data to files, which is very important. There's slides about parameter scans, which we're going to go through. How to run replicate runs. As you know, CompuCell is stochastic. So two runs using the same parameters don't necessarily give the same output. So it's handy to be able to run replicates. And then parameter sensitivity, you take a look at your leisure, which uses parameter scans, though the way I do it is the best way of doing it. But anyway, we're going to skip forward a bit, as they say in Monty Python, to parameter scans. So a parameter scan lets you run replicates and change parameters in your model for each replicate. So it lets you explore parameter values in an automated fashion. So it's easy to do. In addition, it's easy to do multiple runs to explore stochasticity. So you can do a parameter scan on a dummy variable and just say the dummy variable goes to 10 and just do 10 replicates with your base parameter set. Often people think, well, this is a great way to explore parameter space. The problem is this is a combinatorial problem, and the number of simulations you need to do will explode if you do very many parameters. So as an example, if you wanted to scan 10 parameters, which is not an unreasonable number of parameters in a CompuCell model, any biological model, and you say, well, for each parameter, I want to explore 5 values. Let each parameter take on 5 separate values. Well, the number of simulations is the number of values that you're changing raised to the power, excuse me, I got that backwards. The number of values each parameter can have raised to the power of the number of parameters you're doing. So 5 values each for 10 parameters is 10 million simulations. Okay, that's not practical. So to start off, when you're doing pscan, you have to think about how much of a combinatorial explosion you're going to run into. In addition, if you are doing, it's certainly reasonable to do, say, 10,000 simulations. If you're writing screenshots and things like that, you can generate a heck of a lot of data. 10,000 separate simulations, each one saved 1,600 by 600 pixel PNGs. And suddenly you're talking about 3 gigabytes of data that got generated during a run. So something else to worry about. Don't try to do too many, and keep track of how much space it's going to take. Adding a parameter scan to code is a pretty major change. You can remove a parameter scan if you have to, but it's not easy. It's much easier to write a simulation that works and make sure you really like it. It's doing exactly what you want it to do. And then make a copy of that project and do the parameter scan in that copy. So you always have the original unparameter scanned code. Let's see. I think I will see if I can load this project. This is the zip file that I shared earlier, and inside is demo cell sorting base. That's not the one I want. I'm going to do the more complicated one. I'm going to use this one instead. So let me get Twedit running again. Where's my Twedit icon go? The slides go through it in detail. How to add a parameter scan. I'm going to do it live. You'll have this. You can look at the slides after the meeting if you want to, if you don't want to, kind of keep track of exactly everything I'm doing. So I'll go through it manually. Feel free to open up any simulation you want. You can open up the one that I shared, and the one I'm going to do, or cell sorting. Make a real quick cell sorting or something like that. I think what it just crashed. Try it again. But it's loading. But it's open. Can't drag and drop. Looks like there's something weird about this file. Oh, I know what it is. Oh, I closed the wrong folder. I was trying to load out of a zip that's inside of a zip. Which CompuCell can't do. Tried to copy the folder and now I'm trying to figure out where I actually ended up. Finally found it, hopefully. Finally found it. Okay, so it's a generic simulation. This is a cell sorting simulation that logs data as it runs and does various other things. So to add a parameter scan, this already has its parameter scan added to it, but it's reasonably straightforward. Over here in this menu, you right click and you have add parameter scan. And when I click on that, it will create this file. And it will have some dummy values in it. Cat just broke something. It's already got a parameter scan in it, but didn't have the definitions in it.
Jim, open the parameter scan tab.
Oh, you're right. Yep. It's one layer down. So again, add resource, add parameter scan, which creates this tab, and then you have to open that up. And now here's a parameter scan, and this simulation already had a parameter scan defined with it. To add things into the parameter scan, you can go in the XML or your Python, anywhere you want. All right, and you choose a value, numerical value. So I can choose this value here, or I could come back and say, choose this value here. I don't remember if I need the quotes or not, but I can certainly choose this one. Highlight it and then right click, and you get this add to scan option. If I select that, it wants to know, it wants a name, it wants something to call it. So box temp. It needs a numeric range. Let's say we wanted to do 0 to 20 in 2 steps, linear type float. So I've got the domain. If I hit generate, it'll show me the values it's going to do. That's not what I want. What I want is regenerate. So it'll do a temperature of 0, a temperature of 10, a temperature of 20. Okay. It now tells me that it's edited this file, so accept it. Now I've got this new chunk called pots temp. The original value, this is only used if you're going to try to remove the parameter scan. And then the list of values that it's going to do. If you come back and look at where we put it, that number that was there is replaced a curly bracket, the name we gave it, curly bracket, curly bracket. This is a global thing. The pscan code will look at every file for this character string. So you can go onto Python and say print, and if I could type, when the parameter scan is run, before CompuCell starts up, before it does anything, the parameter scan code scans all the files in the project, looks for these name tags, and replaces them with the current value. So when this Python actually runs, this variable name will be gone, and it'll have the value of that string in it. It will also change the value in where we meant it to change back here in the XML. So in this parameter scan, I've already gone through, and what I was scanning was the J value. So the J value, medium to condensing, J from condensing to non-condensing, and then I've got an iteration count for replicates. And I'm going to go back and try something here. Put this back where it was and see if I can remove this from the scan. Yep, removed it from the scan, and it put our original number back. So you can remove a parameter scan. It's just tricky, and it's best not to have to do it to recover your original. So scanning all these values, talk a little bit about iteration count. If you want to do replicates, you can make a dummy variable somewhere. It's called iter count. So iter count is, as I say, just a dummy variable. I'm actually going to output it, but it doesn't really do anything in the code. But the parameter scan will still iterate, step over the values of iter count. So in our steppable, we've told it we want to do 3 times. So for every one of these permutations of my J values, it's going to do 3 simulations, 3 replicates for that particular parameter count. One thing to consider when you're doing a parameter scan is what the heck are you going to look at when you're done? If you turn on saving screenshots, you'll have your screenshots, but that means you would have to go through and look at them by hand. So you have to think about in a parameter scan, what metric, what number are you going to calculate that describes the output of a particular simulation, a particular run, a particular set of parameters. It could be more than one metric. There might be 5 or 6 metrics, but you need some numerical output. In addition, what you'd really like is a table that lists every run, the parameters used for the run, and that output metric. If you're doing cell sorting, you might say, I want to know what the surface contact area is between the condensing cells that are supposed to be in the middle and the medium. If the cell worked correctly, that value will be 0. So what I want to do is write, okay, here's the J values I used, and here's the contact area between the condensing and the medium. So in a finish, I have a steppable that does kind of the setup of parameters, and then in a steppable that just does analyses. It makes a plot window, it sets up some things, but then in the finish function, it opens a file for append, which is actually opened. I don't remember where it's opened. Plot. Oh, here's the file open down here. In the finish, so finish gets called when the simulation stops, when it ends. It makes this string, and you can see our scan variables are in that string. So the parameter scan will rewrite this code so that in the Python code, this double curly bracket item has been replaced with the actual value. And I build up a string that's got all the things that are changing in the simulation, including my iteration count, which is coming from up here. And so I could have just used this directly instead of going through an intermediate variable, though I do need this variable for this to point at. And this value. The self-hetero surface area is calculated in a step. And just here is the code for this particular metric. So that's the metric I chose. So at the end of the run, I will append onto this output file a new line that has all the parameters for the run plus this output metric. So when we open the file, we open it for append, and it just keeps adding on top of it. And there's a little bit of fanciness. It prints a header row, which is going to get replicated over and over and over again, but that's okay. So we've added a parameter scan. We've added some code to calculate a metric. And every time the simulation is run with a particular set of parameters, we're going to log the parameters and an output metric into an output file. So we are ready to do a parameter scan. And I'm going to do some things to make things go faster. I'll do 200 steps per parameter set just so that the simulations run faster. Never use a NeighborOrder. Right? So how do you run it? Well, you run it the same way you do any other run directly from Twedit, though it behaves differently. So you select the file, open in player. And I get a new window. Instead of just going straight to CompuCell, I get this new window. It's asking you.
You
Yep.
may want to save the XML file. I think it's unsaved right now.
You're right. I canceled.
Yeah.
See if I can bail out gracefully. Yes, always remember to hit save.
Yeah.
So again, open in player. We get this intermediate window after a moment. And this is the parameter scan launcher. You can choose whether you're going to save snapshots. I'm going to turn that off just because it creates a ton of output. My default output is to my personal CC3D workspace. I could repoint this to put the output into my project folder if I wanted to. None of these other things have to be filled out. I can say display the command line. Now at this point, I can either, I skipped one. I should talk about this one. This option to use the GUI. If I select use GUI, it'll open up the GUI, open up player, and it'll just do sequential launches and crank through all the simulations. If I turn off the GUI, it'll be a bit faster, assuming graphics are taking a lot of time, but you don't get to see what's going on. You get the command window, but you won't actually get the player window. The third way to run it is I can copy this command and open a command window. I'm on Windows. And paste this command into a command window, and it will start running from the command window. The handy thing about that is pscan is pretty smart, and I can have more than one of these pscans running at the same time. So I can copy this command, open a command window, paste this command, leave that window open another command window, paste the command again, open another command, do it 5 times, and have 5 simulations running at once. And the pscan overarching code will keep track of who's doing what and not duplicate any run. So your 5 windows, the first window does the first set of parameters, second window, the second set of parameters, etc. The only thing you have to worry about is, do you have enough memory to have 5 simulations running at the same time? And do you have enough CPUs? So you probably want to tell the XML that you only want to use 1 CPU. And CompuCell really isn't much of a memory pig, so usually memory is not a huge issue, but it is something you have to think about. So I could have 5 of them, 10 of them, 50 of them running at the same time. If you have a cluster, a compute cluster, you can paste these in as Linux commands and use qsub or nohup or however you want to do it to get a whole bunch of them running on your cluster. But we're not going to do that. We're just going to let it run locally and see how fast it goes. Fingers crossed, I hit run. And now the first simulation's running. I told it only 200 steps, so the first simulation went really fast and closed it out. And here's the second simulation. Now I'm not actually getting cell sorting because the simulations need to run a lot longer than this. But I just wanted to checking to see if the parameter scan is working. It seems working just fine. You might wonder, well, great, where is all the output? And that's going to be a bug that I didn't take into account when I was doing this. There's my workspace folder. So when you're doing a parameter scan, you'll get a workspace folder that says it's the base name of your simulation, which for mine have to have been cell sorting. Get away from that there. If you open that up, see all these sub files. So here's the first scan iteration. It's got everything in it that your regular run would have. If I turned on screenshots, all of my screenshots would be here. And so there's just a folder for every one of the parameter sets. If you open up the files, if I drill down and open up the Python file, Notepad, in the Python file, see, my values have been replaced in the Python itself. So in the Python file, I know what values are actually being used. I can always go back and look at them. But I was writing it to this output file. So I don't need it here, but every one of these projects has all of the replaced values in it. So it's still chewing along. This is the output file I was writing to. Let's see if this is working. It looks like it is. So here's the header row. Here's my so JMC, JCC, JCN. This is the hetero surface, and this is the iteration count. This set of parameters, 835, should appear again. And I don't see it. Maybe we haven't gotten that far yet. Since iter count was the last variable, it's the one that's changing the slowest. If I put it first in the JSON, it would be first, so I'd get, replicate 1, replicate 2, replicate 3. That's not the most usable format. So sort it and get rid of all extra headers. So there's my variables that are changing, result of the simulation, and I'm still on iteration 1 for everybody. Ultimately, I'll get iter 2, iter 3, and I can calculate averages. This set of parameters was run 3 times. What's the average of this value? Could do it. Could do it in the Python, but it'd be a little trickier because you'd have to be reopening the log file and reading it instead of just appending to it. So this will continue to crank along. I can go away and leave and let it keep going. stopping this, when you're running it this way, is stopping a parameter scan is really tricky. It's actually pretty difficult to do. If it was running in the command box, you can just close the command window and it'll stop it. You also Control C and stop it. Here, if I just close one of these, the pscan code is still running, and it just assumes that that means something happened. Actually, it looked like it did stop. I didn't think it would. I think it is. I think it's still running in the background. I'm still seeing Python using a bunch of, yep, there it goes. So even though I killed that window, it just took pscan a minute to figure out that, well, it thinks that the simulation stopped. So it went on to the next one. So running it this way, you have to get brutal. Yeah, killed it with Task Manager. So simulation is stopped. Got all my output folders. I've got however far my log file got. Oh, that's the JSON file. So, oh, here it is. I did get to one iteration two. So this line should be matched 835 is the same as this line up here, 835. The result changed, but I wasn't running a very long simulation. So let's go back to PowerPoint. Getting a little late. So as I say, there's slides here to walk you through, just like I did, how to add the parameter scan. Where's the slide? There it is. This is a discussion of adding an output metric, and then, okay, finally go back, add a parameter scan, assign values, pick a value in your XML or your Python, give it a name, give it a minimum and maximum value, and describe, you want a linear or a log scale. Generate the values just to double check to make sure the values are what you want, hit OK, and that saves it into the parameter scan JSON file. In Twedit, right click on the project and tell it to run, and that'll bring up the dialog. Should be a slide. I guess I don't have it. But it'll bring up the dialog about whether you want to run locally, right now using the player, or save screenshots, run as a command window, etc. Okay, so we got all the way through it, writes output, and we're done. One thing about parameter scans is if I go to Twedit and tell it to run the parameter scan again, it sees that this output file or output folder already exists for the project. And it thinks the scan is done. I could go to Twedit and relaunch the scan, and it hadn't actually finished all of its scans yet. So it would pick up where it left off and keep going. If I get to the point where I've run all the planned scans and I try to launch from Twedit again, it won't launch. It'll come back and say the scan is done. So how do you make it do another scan? Well, the easiest way is that it's doing that based on the existence of this folder. So I can either delete the folder or rename it. And if I rename it, parameter scan can't see it anymore. And the parameter scan, if I launched it again, will start at the beginning and do the whole thing again. So that's parameter scans. There are some samples that already have the parameter scan loaded. If you've got a simple demo, it's worth a quick try of, pick two parameters and give them two values and launch the parameter scan and watch it chew through. One good use of parameter scan is doing replicates where you're not changing any values at all. You just want to do 10 simulations. So you make a dummy iteration variable and say that variable goes 1 to 10, and parameter scanner will automatically run your 10 simulations. Make sure you have some output that's useful and usable and interpretable. Write to a table or something like that, and include your parameters and some output metric that describes how well the simulation did. So I think I will stop there. There are a bunch of demos in the zip file that do various things like writing to the log file and things like that. And I think we will stop there. We already mentioned replicate runs. So is it possible to resume iterations from here after closing it? Yes. If you kill the pscan, if you reissue the same command, it'll pick up where it left off. There's a slight possibility that it might lose one simulation, the one that stopped or crashed. Any computational advantage of using parameter scan from Twedit compared to a manual bash script? Computational, no. It is a big advantage, though, that you don't have to manually take care of changing the values in your script. If you're going to write a bash script or a Python program, for that matter, it would have to know what the parameters are being changed, what their values are supposed to be. It would have to then open up your CompuCell scripts and edit all those files, and then launch the simulation. And pscan basically does all that stuff for you. It is up to you. Yeah, that's an answer. One thing you might use something like a bash script for is if you've got a cluster and you got 256 nodes on your cluster. So what you want to do is have 100 parameter scans running at the same time. Well, you don't want to have to issue the parameter scan at the command line 100 times. So you might write a bash script that, for 1 to 100, launch the parameter scan. And, let your bash or qsub or whatever your batching system is, partition all those jobs out to the various nodes on the cluster. Another thing you can do, and I haven't done it yet, and it would be nice if parameter scan had the option of doing sensitivity analysis. Sensitivity analysis is examining how does the model output change if you make a small change in one parameter. That's the most simple sensitivity analysis. If I change this particular J value and run simulation, how much does my output change? So the way pscan is set up, though, is as an n-dimensional space where it exhaustively visits every combination of the parameters. In a sensitivity, what you want to do is do one run that's the base set of parameters, and then the next run you change exactly one parameter. And then the run after that, you put that first parameter back to where it started and just change exactly one other parameter. So instead of doing n squared or n to the mth power simulations, if you're going to change 5 parameters for sensitivity, you're going to do 6 runs. A reference run and then one run that changes just one value. So it'd be great if the pscan tool could do that, or if there's a way of specifying it that no, I don't want to do 2 values for each of 5 variables. I just want to do one run where this value for this variable changed once, and then set it back to where it was and change the next value once. And typically you change the value 10% or something like that. Look at how the output changes and calculate a sensitivity. How sensitive is the output to that input? It'd be great if pscan could do that, if it was kind of a function of pscan to do sensitivity that way. You can kind of hack it by saying, well, what we're really going to do is, I think I just started pscan. Instead of having an iter count, in addition to having iter count, instead of having these enumerations, we just have a parameter ID number. So if we wanted to do 5 parameters, that would just be a number that goes from 1 to 5. And then we'd have the iter count that tests how many replications to do. And then in the Python, we would figure out what the parameters are supposed to change. So in our Python code, we'd have a line like this here, except for instead of iter count being iter count, it'd be iter count, or parameter count equals the parameter count from the JSON. And based on that parameter count, you would change your values. So I think there's probably a pretty slick way of getting pscan to do all the calculations for a sensitivity analysis. The nice thing about sensitivity analysis compared to parameter scan is you do a hell of a lot less simulations. A parameter scan can be used to do sensitivity. It's just that it'll give you the sensitivity for every individual parameter, every pair of parameters as they're changed, every triple of parameters as they're changed, every possible triple of parameters, every possible triple of 4 parameters. And so you one, doing a lot more simulations than you need, and 2, you got to do a lot of digging to get the single parameter changes out of your whatever your log file is. But it can be done. pscan could be used that way. So I think that finishes mine. As I say, there's a whole bunch of things, a whole bunch of codes in that zip file. Writing the files, doing a parameter scan, using a parameter file so that you can define global parameters and give them units and things like that. I will say then CompuCell 4.7 units have come back to a certain extent, and now there's a parameter in CompuCell for how big do you think 1 pixel is, how many microns do you think it is, how long do you think 1 Monte Carlo step represents. So there's some global variables that have those things, so that if you're going to do a graph in during a simulation, and normally the x-axis would say Monte Carlo steps, you could use that conversion factor and convert it to simulated time and label the axis as simulated time and actually plot simulated time on the axis instead of the rather nebulous Monte Carlo step